Between 58 and 64% of Finnish men carry a Y-chromosome haplogroup that did not originate in Europe. It did not originate near the Baltic. It did not come from the steppe. It did not arrive with the farmers from Anatolia or the hunters who walked into Scandinavia after the ice retreated. It came from Yakutia. That is a region of northeastern Siberia clustered around the Lena River, a place geographically closer to Anchorage, Alaska than to Helsinki, Finland. And if you put a map in front of you right now and drew a straight line from the origin point of this haplogroup to the country where it now dominates the male lineage, that line would cross more of Asia than Europe. Every map you have ever seen of Finnish origins is missing something large, something this specific. Stay with me, because this next part is the reason this video exists. In July 2025, a team led by 2 researchers working with ancient DNA expert David Reich at Harvard Medical School published a study in the journal Nature. They sequenced genome-wide data from 180 ancient Siberians, individuals who had been in the ground for thousands of years, and combined that data with more than 1,000 existing samples covering 11,000 years of human history. What they found placed the ancestors of every Uralic-speaking population, that includes Finns, Estonians, and Hungarians, not near the Ural Mountains where mainstream linguistics had long pointed, but in Yakutia, northeastern Siberia, closer to Alaska than to Finland. Geographically, it's closer to Alaska or Japan than to Finland, said co-lead author Alexander Kim. That is not a rhetorical flourish. That is the coordinate. Now, most of Europe's genetic history fits neatly into 3 ancestral waves: Paleolithic hunter-gatherers who arrived after the last ice age, Neolithic farmers who came from Anatolia around 8,000 years ago, Bronze Age pastoralists who swept in from the Pontic steppe around 4,500 years ago. 3 waves, and the vast majority of Western European ancestry is accounted for. Finland requires a 4th. A Siberian component that runs at somewhere between 10 and 13% across the Finnish genome. And when geneticists plot Finnish DNA on a principal components analysis, the chart that maps all of human genetic diversity onto a 2-dimensional plane, Finland does not cluster with its Scandinavian neighbors. It pulls toward a different axis entirely. It sits alone, pulled east. If you've ever genuinely wondered where you come from, not the family story, but the actual biological answer, subscribe now, because what comes next goes deeper than most people are willing to go. The migration that created that pull didn't just bring a haplogroup, it brought something else with it, something that is still being carried right now in the bodies of living people, and that none of those people chose to inherit. We'll get to that, but first, the people who started the journey, because here is what we don't know about them: their name. Actually, before we go any further, I want to ask you something directly. Drop it in the comments. Where on the map did your family's deepest ancestry begin? And has a DNA test ever placed it somewhere that surprised you? Because honestly, your answer may connect to something I'm going to say in about 15 minutes in a way you won't see coming. We don't know their name. We know where they were buried. We know when they lived. We know what their bones carried. And we know that what their bones carried is, right now, today, in the genomes of millions of living people across Northern Europe. But their name? Gone. No inscription, no account, no surviving word in any language that can be verified as theirs. Bolshoi Oleni Ostrov, Great Reindeer Island. A burial ground on the Kola Peninsula in northwestern Russia, first excavated in the 1920s, 6 individuals, carbon dated to approximately 3,500 years ago, and inside those bones, the oldest confirmed occurrence of Y-chromosomal haplogroup N1C anywhere in Fennoscandia. Theseus Lamnidis and Kertu Majander had been working on exactly this question: the ancient population history of northeastern Europe, the part that the standard 3-wave model kept leaving blank. Their 2018 study in Nature Communications, conducted across teams at the Max Planck Institute for the Science of Human History in Jena, the Max Planck Institute for Evolutionary Anthropology in Leipzig, and the University of Helsinki, pulled genome-wide data from 11 ancient individuals across Finland and the Kola Peninsula. What they expected to find was a gradient, a slow dilution of Siberian ancestry as you moved westward and backward in time. What they actually found was the opposite. The oldest samples had the highest proportion of Siberian ancestry, not less, more. And yet, the data said the exact opposite of what the timeline predicted. These 6 individuals from Bolshoi carried mitochondrial DNA haplogroups Z1, C4, D4 that are today most commonly found in modern Siberia. They also carried a variant in the EDAR gene, RS3827760, linked to hair morphology and tooth shape, found near fixation in East Asian and Native American populations today, and almost entirely absent in Europeans. This variant was in those bones, on the Kola Peninsula, 3,500 years ago. Think about what that means geographically. You are standing on the northwestern tip of Russia, roughly 200 kilometers from the Finnish border, and the genetic signal you are reading belongs to a population whose closest modern relatives live in Siberia and among the indigenous peoples of the Americas. That is not a European story, not by any standard classification that genomics uses. And yet there is no other word for where these people were found. They were in Europe. They were in a burial ground on European soil 3,500 years ago, and their bones carry a story that European population models had no category for. Because nobody had sequenced them yet, the model was built before the evidence existed. They shaped the genetic profile of Northern Europe in ways that are measurable and specific and irreversible. They carried a Y-chromosome haplogroup that now dominates the male lineage of Finland. They left no name, no text, nothing that survived, only the haplogroup, only the genome, only the record their DNA made in the bodies of everyone who came after. That is the most genomically influential nameless population in Northern European prehistory. And the question of where they came from, the actual path that brought them from a starting point in northeastern Siberia to a burial ground on the Kola Peninsula, is a story about something that moved faster across Bronze Age Eurasia than anything the textbooks had accounted for. Somewhere in the Altai-Sian mountain region, around 4,200 years ago, someone cast a bronze spearhead with a snow leopard on the socket. Snow leopards did not live in West Siberia. The nearest habitat for a snow leopard was roughly 1,500 kilometers away. And yet there it is, the figure of a cat that no one in that region had ever seen, cast in bronze, found near the city of Omsk. Because the person who made it had contacts with people who had contacts with people who lived near snow leopards. And those contacts stretched in a single human network across half a continent. This is the Seima-Turbino phenomenon, and it is one of the strangest, most under-discussed events in all of Bronze Age history. In July 2025, when Tian, Chen, Zeng, and Alexander Kim published their Nature study under David Reich's oversight at Harvard, they established something that directly connects to that spearhead: the Yakutia late Neolithic Bronze Age population, the genetic group that the study identifies as the direct ancestors of Uralic speakers, including the ancestors of modern Finns, arrived in the Altai-Sian region approximately 4,200 years ago, and from there they moved west, along the Seima-Turbino network, which means the language that eventually became Finnish and the genetic signal that eventually dominated Finnish male lineage traveled the same route as those bronze artifacts, the same corridor, the same astonishing speed. But here's where it gets strange. The Seima-Turbino individuals the study sequenced were not genetically homogeneous. They were diverse. Some carried the Yakutia ancestry, some carried Indo-Iranian associated pastoralist DNA, some showed hunter-gatherer lineages from different parts of Eurasia. This was not a single ethnic group making a coordinated push across a continent. This was a network, mobile, mixed, multilingual probably, and it moved like nothing else in the ancient world moves. Archaeologists estimate the Seima-Turbino cultural spread covered roughly 5,000 kilometers within what appears to be a single human lifetime's span of transmission. From the Altai to the Baltic. That is not a migration. That is something else. Something that requires a technology and a mobility and a set of social connections that we do not have a standard framework for in this era. And here is the implication that nobody talks about in the popular accounts of this story. If the Yakutia-derived population carried proto-Uralic language westward through this network, and the genomic evidence from the 2025 study strongly supports that reading, then Finnish, as a language, originated not in a forest somewhere near the Baltic, but in the same Siberian river basin that was the genetic ancestor population of Native Americans. The Yakutia LNBA population, the same group. The ancestors of Uralic speakers and the ancestors of some Native American populations trace back to related Siberian groups. Finnish and the languages spoken at the northern tip of the Americas share a common genetic ancestor population. The 2025 data makes that connection explicit. That spearhead with the snow leopard on it was made by someone in a network that carried a language from the edge of the Pacific to the edge of the Atlantic. And inside that language was a genome, and inside that genome was a signal that would eventually sit in Finnish chromosomes for the next 4,000 years, waiting for someone to sequence it. The timeline everyone was using had a problem. That problem has a specific address in Finland, and it is preserved in a lake. The soil in Finland is acid, old acid, bog acid, the kind of acidity that dissolves unburnt bone within a generation in most of the country. Which is why, when researchers go looking for ancient DNA from Finnish soil, they find almost nothing older than 2,000 years. The chemistry takes the evidence. Except at Levenluta. Levenluta is a site in Iso-Kuro in southwestern Finland, today agricultural land, roads, a small lake. During the Iron Age, between roughly 400 and 800 CE, human bodies were placed into what was then a shallow lake or pond. The water chemistry at this particular site, its specific acidity, its cold, its particular composition, preserved bone in a way that almost no other location in Finland manages. Not by design, by geological accident. And what that accident preserved contradicts the name of the province it sits in. Kerttu Majander, working across teams at the Max Planck Institute for the Science of Human History and the University of Helsinki, recovered genome-wide data from 3 Levenluta individuals with enough coverage to analyze. 2 of those 3, along with 6 lower coverage individuals from the site, showed genetic affinity not to the modern Finnish-speaking population of southwestern Finland. Not to the Swedish-speaking population either, to the Saami. The indigenous people who today live predominantly in the northern reaches of Scandinavia, above the Arctic Circle, hundreds of kilometers away. But here's where it gets strange. Linguists working completely independently, studying place name archaeology, not genetics, had already noted that the names around Levenluta trace back to Saami languages, not Finnish ones. The genetic data and the linguistic data arrived at the same conclusion by different routes. The Iron Age population of what is now a Finnish-speaking agricultural lowland was Saami. The people genetically continuous with modern Saami once lived here, in the southwest, in the flatlands, in a place that has been Finnish-speaking for a very long time. Now, hold on, because I want to ask you something before we go further. This idea of ancient populations being replaced by later arrivals, their genetic signal fading but not entirely disappearing. In our last video, we looked at Bronze Age steppe migrations reshaping European identity. What struck you most about that earlier wave of replacement? Drop it in the comments, because what I'm about to show you with the Levenluta data is the same process, quieter, slower, and leaving a specific kind of ghost behind. There was one individual in that burial, laboratory designation JK2065, who did not fit the pattern. Her DNA showed genetic affinity not to the Saami continuous population around her, but to modern Norwegian, Lithuanian, and Icelandic populations. A Western European-affiliated individual submerged in the same lake as Saami-affiliated individuals in Iron Age Finland. She may have been a visitor, a partner from a different community, someone displaced. The genome records her presence, nothing else does. And the Siberian genetic signal, the one that traced from Yakutia through the Altai through the Kola Peninsula, was already in western Finland 1,500 years ago, carried by people who were genetically Saami. And then, over the centuries that followed, it retreated north. The population that carried it was replaced or absorbed. The name of the region shifted from Saami words to Finnish ones. The genetic dominance shifted, but it didn't disappear entirely. That is the point. A lineage that was genomically dominant in southwestern Finland 1,500 years ago is now detectable in the same geography only at low frequency, a faint signal requiring sequencing to find. A ghost that never left. And the bottleneck that followed that displacement, the genetic compression event that created modern Finland's specific gene pool, left something in the bodies of every Finnish person alive today. Something that Levenluta only begins to explain. Population geneticists estimate that the founding event of the Finnish genetic isolate, the bottleneck from which the specific Finnish gene pool descends, involved somewhere between 20 and 40 founding families. Not thousands, not hundreds. The equivalent of a single large village. And from that village, through several thousand years of geographic isolation and limited gene flow from neighboring populations, came 5.5 million people. There is a process in genetics called genetic drift. In a large, diverse, outbred population, harmful genetic variants tend to stay rare. Natural selection catches them, removes them slowly, keeps the population's disease burden relatively distributed. In a very small founding population, drift takes over. Variants that would normally stay uncommon become common simply because they happen to be present in the founders. Not because they are advantageous, not because they were selected for, because there was nobody else's DNA to dilute them. There are three things that the FinnGen study revealed about what the Finnish bottleneck did to the Finnish genome. The third one is the one that changes everything. The FinnGen project, a public-private partnership coordinated by the University of Helsinki, involving 13 international pharmaceutical companies and data from over 300,000 Finnish biobank participants, published its landmark analysis in Nature in 2023. Mark Daly, director of the Institute for Molecular Medicine Finland, was among those who led the interpretation. What the study confirmed was this: Finland experienced not one bottleneck but at least two, one approximately 4,000 years ago and a second approximately 2,000 years ago. And between and after those events, sub-isolates within eastern Finland underwent additional local compressions. Populations within populations, each one compressing the gene pool a little further. The first thing the study found, dozens of loss-of-function variants significantly enriched in Finland compared to all other sequenced populations. Variants that appear at 5 to 10% frequency in Finland and 0.2% globally. The drift didn't just preserve, it amplified. The second, some of those amplified variants are protective. Variants offering some degree of protection against arthrosis, glaucoma, specific forms of heart disease, elevated by the same random mechanism that elevated the harmful ones. The bottleneck was not purely a catastrophe. It was a catastrophe that also, by accident, preserved things. The third thing, and this is the one nobody in a popular account of Finnish genetics spends enough time on. Today, approximately 1 in 5 Finns carries at least one variant associated with what researchers call the Finnish Disease Heritage. 1 in 5. And approximately 1 in 500 Finnish children born today is affected by one of the 36, now 39, officially recognized conditions in that heritage. These are not statistics from centuries ago. 1 in 5. Currently. Today. The bottleneck is not history. It is the current state of the Finnish genome. One of those conditions, congenital nephrotic syndrome of the Finnish type, results from a single founder mutation in a gene called NPHS1. Using linkage disequilibrium analysis, researchers have traced this mutation to a single ancestor carrier living somewhere between 2,000 and 4,000 years ago. One person, one mutation, elevated by drift through the bottleneck, preserved by isolation, carried across generations, and today, a condition that Finnish pediatric units see regularly. The kind of condition that gets used as a teaching case in genetics courses globally because its origin is so traceable, so clean, so undeniable. One carrier, one mutation, 4,000 years. And right now, in Finland, a child is being treated for a disease that started with that one person. In a migration from Siberia. That is the line. The mutations arrived with the founders. They weren't born in Finland. Finland is the amplifier, and the question of who those founders were, where they came from, and exactly when they arrived, is where the next part of this story lives. In the 1960s, a Finnish pediatric neurologist named Reijo Norio started driving. He drove to villages in northern and eastern Finland, remote communities where the roads were poor and the winters lasted 9 months. He wasn't following a genomic hypothesis. There was no such thing as modern genomics yet. He was following the children. Children with diseases that didn't have names in the international medical literature because they had never appeared in sufficient numbers anywhere else in the world to be studied. children in specific valleys, in specific river communities, with specific conditions that their parents had and their grandparents had, and nobody outside those communities had ever cataloged. Norio's work, published in 3 parts in the early 1970s as the Finnish Disease Heritage series, established a framework that the entire field of population genetics would eventually come to use as a canonical example. What he found was not random. The diseases were not scattered evenly across Finland. They clustered, specifically and repeatedly in the east and north, in the communities with the most recent settlement history from the smallest founding groups, in the places furthest from outside gene flow. This isn't just ancient history. These maps, the ones Norio drew by hand, village by village in the 1960s and 70s, are now displayed in medical school curricula across Europe as the clearest visual demonstration we have of how population history becomes clinical reality. His maps are in textbooks. His framework is in research. And if you carry Finnish ancestry, the carrier frequency for at least one of the conditions on those maps sits at approximately 1 in 5. The map is about you. There is a condition called northern epilepsy, progressive epilepsy with mental retardation, sometimes called EPMR. It is a degenerative neurological disease that affects children in specific communities in the Oulanka region of northern Finland. Globally, there are approximately 150 known cases, almost all of them Finnish, concentrated in a single river valley. Because the founder mutation entered that community through a small number of settlers, drifted to locally high frequency, and has been there ever since, invisible to the outside world because outside the outside world, it barely exists. Norio found it by driving to the valley and looking at the children. He was not trying to reconstruct an ancient migration. He had no access to ancient DNA. That technology was decades away. He was simply doing field medicine in isolated communities and noticing patterns that shouldn't statistically be there. But the patterns were there because the migration was there. Because 4,000 years before Norio got in his car, a small group of people carrying specific genetic variants walked into a specific geography and stayed. And their DNA stayed with them. Through every winter, through every generation, through every child born in those valleys. The children were the data. The migration was the cause. Norio connected them without knowing the full depth of what he was connecting. And here is the question that his work raised, and that Finnish genomic medicine is still sitting with. What is the ethical obligation of a population that possesses this kind of self-knowledge? Finland has built a national health registry, a world-class biobank, a universal healthcare system that partly exists as a response to the heritage Norio documented. That infrastructure is the direct descendant of those hand-drawn maps, of those drives to those villages, the biological memory of a near extinction. That is what those maps are. And the people who live in those valleys are not a case study. They are the living inheritors of an event so old it has no name in any language currently spoken. But the event does have a date, and that date connects everything. Let me be precise about what we now know. The Zeng, Kim, and Reich study published in Nature in 2025 dates the westward expansion of the Yakutia-derived proto-Uralic population to approximately 4,000 to 4,200 years ago. The Finnish Disease Heritage bottleneck, confirmed by FinnGen in 2023, is dated to approximately 4,000 years ago. The Lamnidis and Majander study from 2018 places the earliest confirmed N1C in Fennoscandia at Bolshoi Oleni Ostrov, 3,500 years ago, at the trailing edge of the same migration window. And the Finnish Disease Heritage mutations show their highest enrichment in eastern Finland, the region of latest, smallest group settlement, consistent with a westward migration arriving in stages. 4 independent lines of evidence, 4 separate research programs, 1 date, 1 direction, 1 event. Everything I just told you, hold on to it, because it's about to mean something completely different. The standard way to think about the Finnish disease heritage and the Siberian genetic ancestry of Finland is as 2 separate stories. One story is a migration story, a remarkable, genomically verified movement of people from Yakutia through Siberia into Fennoscandia. The other story is a medical story, a set of rare diseases elevated by drift, a population's unfortunate genetic vulnerability, a clinical heritage. They are not 2 stories. The 36 diseases that define Finnish genetic uniqueness are not anomalies. They are the direct biological cost of the migration that made Finland Finnish. Every disease cluster maps onto the bottleneck. Every bottleneck maps onto the arrival, which means the Siberian ancestry that defines Finnish identity, the haplogroup carried by 58 to 64% of Finnish men, the signal that makes Finland sit alone on every European genetic analysis, and the genetic damage encoded in Finnish bodies arrived in the same people at the same moment 4,000 years ago. The founding and the wounding are one event. There is no version of Finnish genetic identity that exists without the bottleneck. There is no version of the Finnish disease heritage that exists without the founding migration. They are not related stories. They are one story, expressed in two vocabularies. One, the vocabulary of haplogroups and principal component analyses. The other, the vocabulary of pediatric medicine and hospital corridors. Take the NPHS1 Finn major mutation, the one traceable to a single carrier ancestor 2,000 to 4,000 years ago. That person existed because a migration happened. That migration happened because a population in Yakutia moved westward, through the Altai, through the West Siberian Plain, to the Kola Peninsula, to Finland. And that mutation, that one variant in that one person, is today a clinical reality in Finnish hospitals. The distance between that ancestor and today's children is the entire length of Finnish history. This is not a tragedy narrative. It is a precision statement about the structure of human population events. Every founding event carries this duality. The identity and the cost are the same act. Finland simply preserved both long enough, through isolation, through the specific chemistry of acid bogs that dissolved almost everything except what needed to survive, for the genomics to make them visible simultaneously. To allow us to look at the migration and the disease in the same frame and recognize that they are the same picture. But here is what that recognition opens, because once you know that the bottleneck and the founding are one event, you have to ask what else that bottleneck preserved. Not just diseases, not just a haplogroup. What else came through that narrow window? The answer to that question is being read right now, in Helsinki, in a study involving 300,000 living Finns. And what it is finding changes the way the bottleneck itself has to be understood. January 2023, Helsinki. The FinnGen study, a public-private consortium involving the University of Helsinki, Finnish National Biobanks, and 13 international pharmaceutical companies, publishes in Nature. Over 300,000 Finnish individuals, the largest coordinated analysis of a single national genome ever conducted at that point. What they found next changed everything they thought they knew about what genetic isolation actually does. The same drift mechanism that elevated disease-causing variants in the Finnish genome also elevated protective ones, variants offering some degree of protection against arthrosis, against glaucoma, against specific forms of heart disease. These variants sit at 5 to 10% frequency in Finland. In the rest of the global population, they sit at 0.2%, too rare to study, too rare to use. In Finland, they are clinically actionable, and at least one of them has become the basis for a drug target currently in pharmaceutical development. Let that land for a second. The same bottleneck that produced 36 rare diseases also preserved a variant that may one day protect people who have no Finnish ancestry at all. Not because anyone planned it, because a small number of people survived a founding event 4,000 years ago, and their specific random genetic snapshot was preserved by isolation for long enough to be useful. Mark Daly, who directs the Institute for Molecular Medicine Finland, described the FinnGen population as uniquely powerful for exactly this reason. The genetic homogeneity that was a vulnerability is simultaneously a scientific instrument. No other population of 5.5 million has undergone this specific compression, this specific isolation, this specific preservation of low-frequency variants at frequencies where they can actually be studied and acted on. The bottleneck is a tragedy and a library at the same time, with no way to separate the two. And then there is this. In 2021, Sini Kermanen and colleagues at the University of Helsinki published a study tracking the fine-scale genetic structure of Finland through the 20th century. Using more than 18,000 Finnish samples, they found something that no one had previously quantified at this resolution. the forced evacuation of 430,000 Karelian Finns during and after World War II, the loss of Karelia to the Soviet Union in 1940, produced a measurable, detectable change in the genetic structure of Finland. A political event, a military defeat, a forced internal migration, encoded in the genome within a single generation, visible in the data today. History writes itself into DNA faster than most people realize. The Karelians who were displaced from their homeland carried sub-isolate specific variants into the rest of Finland, mixing genetic signals that had been geographically stable for generations. The map changed. The genome registered it. The archive is not passive. It records in real time. The FinnGen data has now contributed to COVID-19 host genetics research, to global biobank meta-analysis, to pharmaceutical target identification that benefits populations with no Finnish ancestry. The 40 founding families from 4,000 years ago, the small group who came through the bottleneck, who carried the haplogroup from Yakutia and the disease variants from the founding event, they are, right now, in 2025, contributing to drug development in laboratories across the world. That is the length of that line, from a village in prehistoric Finland to a pharmaceutical lab today. And the line runs through every Finnish genome currently living. But here is the question the archive cannot answer for you, the one the sequencers cannot return with the data. You can order a DNA test online, have it back in 6 weeks. It will give you a category: Northern European, Scandinavian, maybe a percentage from somewhere east, somewhere that surprises you. It will name a haplogroup. It will list an ancestry region. It will not tell you that the signal in that result, the one the test might label Eastern European or Siberian, traces to a founding event that nearly extinguished the people who carried it. It will not tell you that the haplogroup listed in the Y-chromosome section, if you carry N1C, arrived in Europe from the Lena River basin in Yakutia, moved through the Altai with metallurgists casting bronze spearheads with snow leopards on them, crossed the Kola Peninsula in bodies that we found in a burial dated to 3,500 years ago, and entered Finland in a founding population small enough to sit in a single village. The test gives you a category. The genome carries a history the category cannot contain. If you carry Finnish ancestry, or Estonian, or Karelian, or Saami, there is a statistically quantifiable probability, based on current FinnGen data, that you carry a Finnish heritage disease variant in your genome right now. Not a disease, a variant, a signal from a bottleneck that occurred before the Roman Empire existed. The current data puts the carrier frequency at approximately 1 in 5. You may not know, the variant may never express, but it is in your cells, a letter in a word written 4,000 years ago, in a language that no one speaks anymore, by a people whose name was lost before anyone thought to record it. And here is what I want you to hold on to as we leave this. The FinnGen study is ongoing. The ancient DNA database is expanding. If this line of evidence is one you want to follow further, subscribe. The next episode goes deeper into the genomic history you didn't know you were carrying. Finland is not a genetic anomaly. It is a case study in what human survival costs at the biological level, and in what a people preserves involuntarily in the act of enduring. Every isolated population carries this equation. Finland simply left the most complete record. The isolation that made Finland genetically distinct from every other European population is the same isolation that elevated the variants that make some Finnish children sick and that may one day make pharmaceutical treatments available to people who have never set foot in Scandinavia. The library was built by the catastrophe. The identity and the cost arrived together. And the people who created both of those things never knew they were doing either one. The people who walked out of Yakutia 4,500 years ago, carrying a language and a haplogroup and a cluster of rare mutations that would eventually be named a disease heritage. They did not know they were founding Finland. They were surviving winter, and their survival is in your genome.